Good afternoon. Thank you all for joining us for the virtual 9th Annual Forensic Science Symposium Lunch and Learn poster session, hosted by the Global Forensic and Justice Center. I'm very happy to introduce Ms. Amber MacInnis, PhD student at Florida International University, Department of Biological Sciences, with her presentation, Competition Among Three Species of Forensically Important Blowfly Species. Welcome, Amber.
Thank you very much. And so I am going to be talking about my research that I did. This was part of my master's work that was conducted at the University of Nebraska-Lincoln that I finished up in 2018. So what I was looking at, and I was interested in the arrival patterns of insects on carrion. So we know that there's a succession pattern to these. But I was interested in trying to figure out some reasons why we might see the patterns we see, because in forensic entomology, usually the first insect species to arrive are the blowflies. And then even amongst there, the first blowfly species to arrive is considered the most forensically important species. And so I was curious on why we see certain species arriving first pretty much all the time versus some species that are arriving later. And one kind of mode that I thought might, and has been posed in the literature that I thought might be interested in looking at was competition. Because if you look at the competitive exclusion principle, it states that two species cannot occupy the exact same niche. And so these species are all occupying a carcass, and so they have to have some different maybe arrival strategies or seasonality to them in order to avoid this competition on the carcass. And so what I did was I decided to use these replacement series models. And so what it is I have, I used three species in this study. I used Formia regina, which is the black blowfly. Typically it is known to be a later arriver and it prefers fall and spring temperatures. I used Lucilia serracata, which is known to be an earlier arriver and tends to prefer the warmer summer temperatures. And then the last species I chose was an interesting species, which is Chrysomia rufifacies. It's actually an invasive species that has come over from Australia in the 1980s. It was first recorded in Texas. And what they do is they are a later arriver. They're more of a warmer weather fly, but they tend to be a predator on other maggot species. So once they reach a certain developmental stage, they'll eat the other soft-bodied maggots. They're called the hairy maggot blowfly because they have a bunch of spines on them, which help them they wrap around their prey, and then they can eat them that way. And so each one of my species has different life history traits. And so I set up these competitive scenarios. So I placed 20 maggots inside of a mouse. The mouse is about 5 to 7 grams. If you have snakes, these are fuzzies. It's the term that they use for there. And I placed them in different ratios for each one of these replacement series models. So the first one, my treatment 1, which you can also see on figure 1, it was a 1-to-0 ratio. So all of species A, so 20 of species A and 0 of species B. The next one is a 3-to-1, so it would have 15 of species A, 5 of species B, a 1-to-1 ratio, so 10 and 10, and then we go to 5 and 15, and then 0 and 20. And so what I did is I had those treatments set up, and there were 10 replicates for each one. And then I just let the species, they were all placed on as newly hatched larvae at the same time, and they were just stored in a growth chamber, and then once the adults emerged, then I was able to record the species and develop these tables that you see here, figures 1 and 2 over there on the right side of the poster. And so I plotted the number of species, the average number of species across all 10 treatments that were recorded for each one of the different ratios. So you can see in this case, in figure 1, the solid line is Formia, the dashed line is Lucilia serracata. And so the intersection of that graph, whether or not it's shifted, if it were a perfect no competition scenario, the intersection would be found directly over the 1-to-1 ratio, meaning both lines are, increasing or decreasing at the same rate. So a shift, in this case in figure 1 to the right, indicates that Formia might have some kind of competitive advantage. In figure 2, you can see it shifted to the left, meaning Chrysomia rufifacies might have some kind of competitive advantage. There is not a replacement series model for Formia versus Chrysomia rufifacies, because Chrysomia rufifacies ate all of the Formia in these treatments. There were no survivors. But in addition to, I wanted to have some kind of numerical calculation. So I used the RCC, which is the relative crowding coefficient, and then the modified relative crowding coefficient in order to determine competition. So a value of 1 would indicate no competition. A value that is significantly different from 1 indicates competition. And so what I found is there was significant competition between Formia vagina and Lucilia serracata, with Formia being the superior competitor. That may have resulted in, so Lucilia is an earlier arriver, so it may be one of those that Lucilia selected for this early exploitation because Formia may have been a superior competitor in the past. I also found no significant competition between Lucilia serracata and Chrysomia rufifacies, even though the intersection is shifted. And that's because one of the reasons that we're thought to have not seen that is Lucilia is known to overlap with Chrysomia rufifacies. They're both warmer weather species, so they can be found on there at the same time. And what's been shown in the literature is that when Lucilia serracata encounters Chrysomia rufifacies, they tend to form groups away from them to help avoid the predation. They might even leave the carcass earlier to help avoid that predation. So seeing no significant competition there shows that this may be they're better adapted to deal with this predator than a species like Formia that never overlaps with them and was completely consumed in those studies. So that's, I guess, yeah, that's what I did. And I will take any questions that anyone has.
We have a question. How did you make your choice of species for this research?
The species that I chose are all common species that are found in forensic casework, with Formia regina being one of the most common species found in forensic casework. Lucilia serracata is common, and then Chrysomia rufifacies is common in casework as well. And then they all have different life history traits, which is what was interesting about choosing the different species. I wanted to choose these life history traits just so if we could kind of examine if, you know, why we see these life history traits is because of some kind of competitive differences between them.
Excellent, thank you. As a lay person, I have a question. Is there a scientific notation for when one species completely devours the other species during a test?
I've gone back and forth with my advisor on this. I use the term predation. Is the term that I use. Like, they are preying upon those species. And so that's, I think we agreed on we could use the term predation for there.
That sounds a lot better than species A was far more tasty than species 2, or...
Yeah, and they may be equally tasty. It's just, you know, the Lucilia species is just better adapted to dealing with it because they've dealt with that species, you know, for how many years, and, you know, they've kind of just evolved to, like, say, hey, we're getting kind of out of town when we see you because, you know, we're tasty. And it might also work in Lucilia's advantage because if, you know, Chrysomia rufifacies prefers Formia over Lucilia, then they're also eating another competitor for Lucilia. They're like, you can just take out one of our other competitors. And so, yeah, it might kind of be a win-win for Lucilia in this scenario.
It's fascinating competitive behaviors in such similar species.
It is, like, this was really interesting to examine.
Do you anticipate furthering this research by expanding to other species?
I would love to expand the research. I don't think it's on my plan simply because I have moved on from my master's and I've already started work on a different project in my PhD. But I would love to expand to other species or even expand this project into, instead of placing species on at the same time, incorporating that delay that we see with such species. So now we know that Formia delays and Lucilia is early. So if we put Lucilia on 24 hours before Formia, examining that interaction to see if it still has a competitive advantage or if that competitive advantage is erased because they have been delayed. So I think the research, I think it's an interesting area for someone to go. I just don't think that it's something that I'm going to pursue at this time.
If someone were interested in continuing the research, your correspondence email is on the poster, correct?
Correct, yes. So if anyone wanted to contact me for more information on this, my email's on the poster. Feel free to let me know.
Amber, do you have any theories on how the competition between the species may affect using this blowfly as an indicator of time of death for forensic purposes?
So one of my my theories would be more associated with the Chrysomia rufifacies and Formia. And so what we've seen with Chrysomia rufifacies, even here in South Florida and Miami, the species is very prevalent. They're thought to be a warmer weather species, but when we actually see them most commonly when we're placing out traps and stuff like that, is we see them more toward the winter time after we've had our first cold snap. And there's even literature that supports the adults have been found active in 10 degrees C, or between 9 and 10 degrees C. And so this species is expanding even into the United States, up into Indiana, Michigan, where it is having cooler weather. And so potentially they could interact with Formia. And so it is one of those things that both species delay. And so it could, I can't remember off the top of my head right now who has the faster development of the two species. I can't remember which species has the faster development. But if, say Formia were to have a faster development than Chrysomia rufifacies, you'd want to use them for casework. And if they're all consumed by the rufifacies, then it could have maybe some kind of effects on that case. So Formia is either going to have to figure out how to adapt or become locally extinct, potentially in some certain areas as an extreme.
It would be interesting to track the incidence of the different species from different body farms geographically, Tennessee versus Florida versus somewhere further north.
Right, and even with casework, it could be recorded with casework if we're seeing these two species that are found together, or through people trapping and things like that, if they are found together at the same time. Because just based on this, when they were in this scenario, they didn't fare well. I mean, out of, I have 10 trials of this and I had no survivors for any of that, which is compared to Lucilia when they were placed with rufifacies, I had about 50% survival. So it's significantly decreased between the species. So
that sounds like an excellent arm of research to be added onto with anybody who's interested.
I agree. I think there's so much that could still be done in this that it's really interesting, but I only had the short amount of time to work on it. And so that's why I'm like, anyone else that wants to take it is, I've opened a door here with the beginnings of the research.
That's great. Thank you so much for that. As a reminder to our participants and anyone who may have come in during our discussion, please use the group chat for posting your questions, and I will make sure that I read them to our presenter to answer. So Amber, do you remember what it was about this particular field that interested you in structuring this research, this poster?
It's simply just being out where I started some of my work out in Indiana and just seeing the different species that come when you leave things out, the different seasons when things are found, just kind of when I was learning kind of forensic entomology, just why do we see this behavior? Because there has to be a reason for the behavior. And so I was just more interested in like okay, we see the behavior and I understand it, but I just wanted to kind of, I don't know, I thought it was interesting to figure out the why type reasons of what we were seeing. Because I mean, all of this fascinates me. I mean, these species fascinate me, their behaviors fascinate me. I'm especially fascinated by Chrysomia rufifacies. I just think they're awesome.
What are the characteristics of that you find so interesting?
That they just eat anyone that gets in their way.
That
I
is
love that.
certainly an evolutionary advantage.
Yeah, someone's in their way, someone's taking up their resource, they just eat them. And it's like, that's awesome. Like, more power to you guys. Because there's really not a lot of blowfly species that act in that fashion. So
We have a question for the chat, and I apologize in advance for my pronunciation. I am a layperson, not a scientist. I will do my best. The question is, do you have an idea on how Chrysomia rufifacies behaves when it interacts with species of different families, such as Sarcophagidae? For example, in Costa Rica, we have noticed that several species of Calophoridae may have been displaced by rufifacies, but that may not be the case with species of Sarcophagidae.
I don't have any knowledge, just because that's not a species that I did. But I can say just from the like field research that I have seen when I've like placed out deer and things like that, that if I have seen Chrysomia rufifacies on Carrion, it's typically been the most predominant species. Whether that's by, forcing things out or eating other things. So I think it's potentially, I mean, just seeing how the behavior of one species that interacts with it versus one that doesn't. I could imagine that, I know the sarcophagid larvae they typically tend to be larger. And they do have the advantage of that they are laid as larvae instead of laid as eggs. So they do start as some kind of advantage where they might be a little bit larger. But I would think that they would still be subject to potentially some predation by them, and could be potentially forced out by them. The only experience I've had in the past where I have used a predaceous larvae, it wasn't Chrysomia rufifacies in this case, it was a sister species, Chrysomia albiceps, when I was over doing some research out of the country. I did have sarcophagid larvae and I didn't even chance it. If I had them on a cups, I removed all of the predaceous larvae out. Just so that they couldn't eat it, because we were worried about them consuming the larvae.
Amber, I see in the write-up on your poster in the very bottom paragraph a mention of how climate change may affect the area of activity for these different species. I assume that would also be a subject of further research, tracking the appearance possibly by case studies or observations, how species move around the country versus historic location data.
Yes, that would be something that you would need some kind of just people continuing to make observations. I know even with when we just talked about this the other day, when Chrysomia rufifacies was introduced in the United States and even in South Florida, if you compare South Florida's species that are common now compared to the historic species before its introduction, they are different. And so, I mean, that's not a climate change per se, but it is forcing species out. And so if, the weather, is warming up, so Chrysomia rufifacies potentially could expand for longer periods of time in other parts, of the country, then they could be forcing out some species that were once common, and they could make them kind of a more obsolete species. So you would see them less commonly. And so just with constant, just people continuing to make observations, we can kind of examine any of these trends that might kind of emerge over, years.
Especially with the heavy predation by the species, I would imagine.
Yes, yes, because they have been, I mean, been shown to change the species composition for an area, um, just because they can be a superior competitor and force other people out. So.
Amber, for those of our participants who just came in, would you mind summing up your highlights and conclusions from this research?
Yes. So what I was looking at is I was looking at competition as a way to explain behavioral traits that we see with forensic species in terms of arrival times and species or seasonality differences. And so what I did was I developed these replacement series models where I varied the number of larvae that were placed. So I had 5 treatments, each with varying number of larvae, and I paired them against each other in a group of in pairs. So I had 3 species that I kind of paired off everyone with the other. And what I found was that I found significant competition. So between the species Formia regina, which you can see on figure 1 on the right, over Lucilia serracata. So you can see the intersection of that graph is shifted over to the right in favor of a competitive advantage for Formia regina. And I also had a significant result from the relative crowding coefficient that was calculated. And so in this case, it was significantly different from 1, which would indicate no competition if it were 1. But because it was significantly different, it indicates a competitive advantage. One of these, Lucilia serracata, tends to arrive early on Carrion, versus Formia tends to delay oviposition on Carrion. So it may be that Lucilia serracata selected for early oviposition on a corpse just simply to avoid this competition by Formia, since they do have that competitive advantage. The next thing that I found was that Lucilia serracata, and between Chrysomia rufifacies, there was no competitive advantage amongst either species. So Chrysomia rufifacies is known to be a predator on other maggot species, but these 2 species overlap in distribution very commonly. They're both warm weather species, and so the Chrysomia rufifacies, though, were not able to fully prey upon all of the Lucilia serracata larvae, because it's been shown in the literature that they tend to form groups away from them, or even leave the carcasses early in the presence of the species, because they will prey upon them. And then the RCC and RCCM value, neither one showed a significant difference from 1. So there was, even though that graph is shifted a little bit to the left in favor of Chrysomia rufifacies, there was no significant competitive advantage. It might be minor at best. And then in the last series that I set up with Chrysomia rufifacies and Formia regina, I do not have a figure for that, simply because all of the Formia were consumed during this, leading, full predation of Chrysomia rufifacies on Formia regina, which is kind of an ultimate competitive advantage there. And those 2 species don't overlap in distribution at all. So it's an indication of, Chrysomia rufifacies is supposed to be warm, Formia is supposed to be cool, but Chrysomia rufifacies has been found expanding farther north in the United States at cooler temperatures. And so it may be that these species will overlap one day, causing localized extinction potentially as a kind of extreme for Formia regina, unless they were to adapt to their competition. So.
Amber, have you heard any feedback from researchers working along the same lines of inquiry, or just with other blowfly species regarding your research?
I haven't had any feedback. I am actually awaiting feedback. My manuscript has been submitted for publication now, so I'm hoping that it was sent to people that are doing the same type of competition experiments. I haven't presented this research a lot as of yet. And so I have heard some competition stuff when I've been at other forensic talks. And so I have seen work that other people are doing in terms of competition. So I'm hoping that with the reviewer comments on my manuscript that I will get some kind of feedback from people that are doing this in the field.
Congratulations on your submission. It's very exciting that you can present this here for us.
Yeah, thank you. Thank you. I am it'll be my first one, so I'm really excited.
And what is the topic of your PhD?
I am looking at marking transgenic insects. So I'm inserting genes for fluorescence into blowfly species so that we can mark them and we can do some validation experiments with determining time of death or using the maggots to determine time of death. So completely different from this.
Fascinating, though. Would the fluorescence gene, or what, excuse me, would the fluorescence work under a certain wavelength for excitation, or is it more of a luminosity generator like jellyfish or microalgae?
No, it'll have a certain wavelength for excitation for it. And so, yeah, and it's heat induced. So it's under a heat shock promoter. So it won't start producing the protein until they're warmed up from, whatever temperature you've been rearing them at. So I have to heat shock them first. So it'll be interesting. It's going to be fun.
That's fascinating. We have another question from the chat. Impressive work. Out of curiosity, what would be the competition status if all the 3 species were kept together? Is 20 larvae enough to study this phenomenon? Did you check the effect of environmental parameters as well? I'm sorry, that's 3 questions. Right. Just the first one, out of curiosity, what would be the competition status if all the 3 species were kept together?
So this is an interesting one to think about. If all 3 of the species were kept together, what I would expect to see is Chrysomia rufofacies coming out with a top advantage here. And then I think Formia might actually be the worst off in this scenario because Chrysomia is shown to obviously have more of a preference and an ability to consume Formia larvae. And so that also works in the favor of Lucilia because now one of their competitors is completely eaten from this study. And so I think if all 3 species were together, we might not actually see any differences we might not see any differences than what we saw with the Serracata and the rufofacies cross in figure 2, especially if Chrysomia was able to consume all of the Formia larvae again, because the behavioral traits wouldn't change for Lucilia. And so Formia would still kind of have no idea what to do when they're bumped in by one of these spiny maggots. They would just not have any idea, versus Lucilia is like, yep, getting out. So it would be interesting to see, though, if you were to put all 3 of these species together.
Thank you. The second question is 20 larvae enough to study this phenomenon? This might be a statistical question or a relative crowding coefficient.
The reason I chose 20 here is because we wanted to avoid any effects from the maggot mass effect. So we wanted to avoid the heat effect for development and all those things. And so it has been shown that 20 larvae is, significant or is enough to avoid that effect. It has been used in other studies as a number before as well for like development studies. And then the mouse that was used is enough. The weight of the mouse and everything was enough to sustain a mass of 20. If you were going to do anything larger than 20, now you start talking about potential other variables like the heat effect from maggots and masses. And so it would get a little more confusing, which is why we kind of stuck with 20 there.
And the third section, or the third question, is did you check the effect of environmental parameters as well? I assume that would include the temperature raised by larval mass.
Right. And so since we had such a small number, we didn't get any of that larval temperature increase, and they were under the same conditions across. They were in incubators or in growth chambers. They were all kept at 25 degrees. They were on the same light cycle throughout all of this. So everything was held as consistent as possible. The mice were in that same weight range. The mice were treated the same way before the larvae were placed. The larvae were all placed in the same way. So hopefully we kind of minimize any of those environmental effects, but it would be one thing that potentially, if you were to do this in a more like naturalized setting, you might see some environmental effects come into play. But we weren't able to examine any for this study.
So this was conducted in a laboratory, not in a field environment.
Yes, correct. This was all done in lab.
Thank you. Another question from the chat. Are there any reports that indicate that Chrysomia rufifacies is already competing and displacing Formia regina or any other species of blowfly that you know of?
I know that I've talked, and I'm sorry, I don't know the references off the top of my head for this. We talked about this the other day with my current advisor, that if you were to compare the commonly found flies in South Florida, where we are now, and I think even in Brazil, which Brazil doesn't have Chrysomia rufifacies, they have a sister species that's also predaceous, but we'll stick with South Florida. If you were to compare the commonly found blowflies, they are different than what we found after the species has been introduced. There have been a couple introduced species here into South Florida, so it's hard to say whether or not that effect was simply because of Chrysomia rufifacies or a combination of the introduced species that are here, but the flies that used to be common are no longer commonly found here in South Florida. So that could be an effect from this thing. And I've also noticed just when I've had Chrysomia rufifacies on Carrion, they tend to be a predominant species. I may find very few of any soft-bodied maggots or any of the smooth-bodied maggots, meaning the other species. But I find predominantly Chrysomia rufifacies, and so that could be something they not necessarily displacing over the whole area, although they might be, but displacing on a carcass is certainly possible.
Thank you. And what is the general range of your field observations lately?
So I've done, yeah, I've done observations. Most of my observations are coming across from Indiana, where I was going to school when I started kind of all forensic entomology. And so we had the opportunity that I worked with a forensic entomologist there, Dr. Neil Haskell, and he had a farm where we were able to place out anything that, anytime we wanted, we could place stuff out. And we pretty much always had something going for just kind of even observations for us, just kind of learning and all of that. So that's where a lot of my observations come from. I have seen Chrysomia rufifacies also here in South Florida. When we kind of buy like a rotisserie chicken, just like an uncooked one, and place it out. And then I've seen them in South Dakota as well. I've done some, and again, they were the predominant species as well that I found.
Amber, as we're wrapping up today's session, thank you for your time, first of all, and your expertise in answering the questions. Would you mind giving us the summary of this research before we leave?
Yeah, no problem at all. Thank you very much. So again, what I aimed to do in this study was to see if I could explain the successional patterns that we see with blowflies on Carrion by examining competition. And so that's one of the methods that we have thought that might explain this. And so what I found throughout my study is I found that there was a significant competitive advantage when Formia regina and Lucilia serracata were placed in the same treatment, possibly leading to the idea that Lucilia serracata may have selected for earlier exploitation of this carcass in order to avoid the competition, because they are outcompeted by those. Another thing that I found was that there was no competitive advantage between either species of Chrysomia rufifacies or Lucilia serracata when they were placed in the same group. It's kind of surprising because Chrysomia rufifacies is a known predator on other maggot species. However, Lucilia serracata and Chrysomia rufifacies overlap in distribution. And so through the literature, it's been shown that Lucilius arracata forms masses away from Chrysomia rufifasces when they've been in contact with them. And then the last thing we found was the last species, or the last pairing we had, was Chrysomia rufifasces and Formia regina. And in that case, all of the Formia regina were consumed by the predator species Chrysomia rufifasces, leading to kind of the ultimate competitive advantage here. And those species don't overlap in distribution, although Chrysomia rufifasces is being found in cooler weather. And so it potentially will overlap with Formia regina, or could overlap one day, leading to possible localized extinction as kind of an extreme in certain locations. And so this kind of shows that, or hopefully it's kind of the start to showing that competition might explain these different successional patterns that we see between early arrivers and later arrivers on Carrion.
Thank you. This is fascinating behavioral and observational research. I appreciate your presentation.
Thank you very much.